Welcome back to MicroVerse. In the previous lecture, we learned how bacteria move using flagella. But movement alone is not enough. Once a bacteria reaches a suitable surface or a host cell, it often needs to attach firmly. This attachment is important for colonization and in pathogenic bacteria may contribute to virulence. So today's question is, how do bacteria attach? The answer involves another group of a bacterial surface appendage, which is pili and fimbriae. So, what are pili and fimbriae? Pili or fimbriae are short, fine hair-like surface appendage of bacteria. They are thinner than flagella, shorter than flagella, then straight rather than helical. Also, made a protein called pilin, which is not primarily involved in locomotion. A bacterium may possess numerous fimbriae over its surface. Their major roles can be broadly understood as a adhesion or genes transfer. So the first and most important type is common pili or fimbriae. These are usually short, fine, numerous, and distributed over the bacterial surface. Their major function is adhesion. So the fimbriae help bacteria attach firmly to the host epithelial cells, other cells, and various surfaces. This attachment helps bacteria remain at a favorable site instead of simply being removed by mechanical forces. So, these three terms describe the sequential stages of a bacterial pathogenesis. So, step by step process of how bacteria attach to the host and potentially make them sick. So the attachment is important for colonization, and after colonization, pathogenic bacteria may contribute to virulence. So remember this flow: first attachment, then colonization, and then possible infection happens. So this is why fimbriae can contribute a bacterial virulence. So here we see the clinical importance. Imagine bacteria entering the urinary tract or respiratory tract. The body has mechanism to remove microorganisms. For bacteria to establish themselves, they often need to adhere to host cell. Fimbriae act as an organ of adhesion. So this is the key concept. Flagella help bacteria move, then fimbriae help bacteria attach. So do not confuse the two. Then are pili involved in motility? So here is an important correction to common misconception. Fimbriae or common pili are not organ of locomotion. They may be present in both motile bacteria and in non-motile bacteria. So the presence of pili does not mean that bacterium can move. Then structure and composition. Pili are composed of repeating protein subunits called pilin. This structure originates from the bacterial cell membrane and project outwards as a fine surface filament. Because they are extremely small, fimbriae are generally not seen with an ordinary light microscope. For direct visualization, electron microscopy is used. The next sex pili. Now let's move to a special type, sex pili, also called F pili or fertility pili. So these differ from common fimbriae. They are generally longer and fewer in number. Their major role in is not ordinary addition to the host cell. Instead, they are involved in bacterial conjugation. So, what happens during conjugation? A donor bacterium possessing the appropriate fertility factor can form a sex pilus. The sex pilus establish contact with the recipient bacterium. This allow the transfer of genetic material during conjugation. Then sex pili and conjugation. Now, unlike common fimbriae, sex pili have a completely different role. Imagine two bacterial cells. One is donor bacterium carrying the F factor, also called the fertility factor. The donor produces a sex pilus which helps it establish contact with another bacterium, which is the recipient bacterium. Once the bacteria come into close contact, a connection is established between them, allowing genetic material to be transferred from the donor to the recipient. This process is called bacterial conjugation. So, in simple terms, donor makes contact, then connection between bacteria and form genetic material transfer to recipient. The important point is that the F factor contain the genetic information required for fertility and for the formation of structures involved in conjugation, including the sex pairs. So here we see the difference between common pili and sex pili. So now don't confuse common pili with sex pili. Common pili or fimbriae are usually short and numerous. Their main function is adhesion. They help bacteria attach to the host cell or other surfaces, which is important for colonization. Then the sex pili, on the other hand, are longer and fewer in number. Their main role is not ordinary attachment to the host cell. They help establish contact between the bacterial cells during conjugation and allowing genetic material to be transferred. So remember, common pili help bacteria attach to the surface and sex pili help one bacterium connect with another for genetic transfer. So next, surface pellicle formation. So fimbriae can also help bacteria adhere at the surfaces. So fimbriated bacteria may form a surface pellicle in liquid culture. This can serve as an indirect indication of the presence of fimbriae. So fimbriae help bacteria attach to the surface. When many fimbriated bacteria grow in liquid medium, they can adhere to each other and to the surface or interface, producing a visible layer called a surface pellicle. So, in short, you remember this flow. Fimbriae help bacteria stay, then attached bacteria accumulate, then a surface pellicle may form. The next, hemagglutination. Some fimbriated bacteria can attach to red blood cells and produce hemagglutination. This property can used as an indirect method for detecting certain fimbriae. An important certain fimbriae manage sensitive hemagglutination. In certain bacteria, the hemagglutination caused by fimbrial adhesion can be initiated by D-mannose. This is known as mannose sensitive hemagglutination. Then detection of a fimbriae. Fimbriae are very thin, therefore direct demonstration is mainly by electron microscopy. Indirect evidence may include surface pellicle formation and hemagglutination in appropriate fimbriated bacteria. These are useful microbiological examination. Then if asked, differentiate flagella from fimbriae. Remember, flagella is longer, thicker, and responsible for motility, while fimbriae is shorter, thinner, and mainly responsible for adhesion. And the sex pili is mainly conjugation and genetic transfer. Then here I give you a mnemonic PILI. P for protein pilin, I for interaction and adhesion, L for link bacteria during conjugation, and I for important in colonization. To remember the major importance of pili, think of four ideas. Pilin is the structural protein. Common pili are involved in interaction and adhesion. Then specialized sex pili link bacteria during conjugation and adhesion help the bacterial colonization. So the simplest distinction is flagella move, fimbriae attached, and sex pili transfer genes. So, pili and fimbriae are not simply tiny hairs on the bacterial surface. They can play important role in adhesion, colonization, virulence, surface pellicle formation, hemagglutination, and genetic transfer through conjugation. The key idea is a bacterium may use flagella to reach locomotion, but fimbriae help it stay there. And spatial type, the sex pillars allow bacteria to establish contact for genetic transfer. So we have now covered the major external appendages involved in movement and attachment. So next major structure in our sequence is how can bacteria survive extreme condition. So thank you for the learning with MicroVerse. Let's master microbiology, one concept at a time.